This is Aubrey Warner, NSH's Director for Meetings and Education. Travel restrictions around the coronavirus led NSH to offer our annual convention virtually this year. I was heartbroken to not see our attendees in person, but really proud of the great education histology leaders and NSH members produced, including one of my favorite features of our convention, our scientific poster gallery. Please enjoy this year's installment of Poster Podcast featuring conversations with our 2020 poster presenters discussing their research. Thanks for listening.
Hi, this is Mary Abule, and I am speaking with Damien Laudier. I hope I have your name right, Damien. And your poster is entitled High Resolution Histological Preparation of Araneomorphae and Mygalomorphae chelicerae using a modified petrographic technique. And that was a challenge for me to learn the title. It is very interesting. You are, I guess, an entomologist, from what I understand, and I assume you don't mind talking about spiders, even though I don't like spiders. But you are finding some beautiful ways to show the arthropods as they are. And I find your poster fascinating. Is this your first poster?
No, this is not my first poster. I've done several posters. Most of my posters have focused on arthropod histologic technique.
Okay.
In fact, I think all of them have, now that I'm going back in my head. I do give workshops as well, and I've done workshops on arthropod histological technique, but I find that the more popular ones are actually the ones that I do on bone histology.
Okay.
So I tend to save the posters for the arthropods.
Very nice. I know that you won the Jules Elias Excellence in IHC Award in 2019. So you must have a lot of very interesting and difficult kind of techniques for showing a lot of the bone and the arthropods that you've shown. Just really gorgeous photos, and your photography must be amazing for all the different kinds of things that you're doing. But your technique that you show in the poster is, a basic resin technique, but such beautiful staining that you have shown. And why is this topic something that was important for you to show?
Well, it's important for me to show, and I get asked this question a lot because I work in such an arcane, esoteric area of histology. Fortunately, there are people out there that feel that biodiversity, species diversification is important, and looking at the microstructure of a lot of species that just tend not to be studied by a lot of people. There are probably, I think, 40,000 different species of spiders. And if you think about that number versus mammals, I don't have the figure top of my head, but it's probably maybe, I think, 4,000 to 5,000 different species of mammals. I guess I'd have to look it up, but the number is not close at all.
No, it's not.
So, most histologists do work with vertebrates, so whether they're in the clinical laboratory working with human samples or in a veterinary environment, a research environment where you're talking about more mice and rats. So that's really what's familiar, and all of those techniques are well documented. You can pick up any histology technique book and find a protocol that would be applicable. Where something with insects or in their relatives, such as spiders, for instance, a lot of the techniques are not applicable. They don't carry over. They don't cross over well. They just don't work in the same way. And when you're talking about something like chelicerae, and I can explain what chelicerae and chelicerata and all that means if you'd like for me to do that. It's probably going to go longer than 15 minutes.
Well, that's all right.
But spiders, the chelicerae are fangs, and these are the apparatus that are used to deliver venom into to their prey. These are very dense, hard, chitinous structures with some other elements involved that really give them a hardness that cannot be overcome with traditional techniques. So certainly something like a decalcification solution is not going to work. Other softening agents that may be employed can be damaging to the structure and kind of defeat the whole purpose of looking at them histologically. So the point of this poster, obviously, there's something to, like I said, to understand the world around us in terms of the creatures that inhabit the planet with us, but also how they work, looking at the beauty of their tissue structure.
Sure.
So, like I said, it's important is a relative term. It's important to me. It's important to a lot of other people, fortunately. So that's how this poster came to be about. So I'd like for other people to appreciate a different type of technique, a different way of looking at things, and also to learn something new about something that a creature that many people like yourself may not have so much appreciation for.
Well, yeah, I have to say, when I first looked at the first couple of pictures that you have there, are the fangs of the spider, and it was rather chilling, but I think it was very interesting, and the photographs are incredible. It's very, very interesting to see the makeup of, as you say, the chitinous material of the fangs and how you display them so beautifully with just, toluidine blue and trichrome stain. Very, very interesting. What is the first thing that you'd like people to notice when they look at your poster?
Well, I'd like people to notice, I guess, verbiage, the word. So many people may not know, like, some of the words that you struggled with, and probably said, well, what is that? What are the differences between the two types of spiders, like Araneomorphae and Mygalomorphae? What is that? What does that mean? What are chelicerae? So I'd like for people to, like, oh, let me, if I have my phone, let me take out my phone and Google that, see what that actually means, and then get into, once they understand what is going on, then get into the images and the technique. And so get into the vocabulary and then dive into the poster and see how different it is from what most histo techs are doing day to day, which is the paraffin processing method.
Right. No, it's I too Googled, so I do know the difference between the two. It's how their fangs are positioned, and that kind of basically is the difference between them. But a very, very interesting topic, and something that most histotechs probably would not do. But like you had mentioned, it kind of is a good technique for bone, in addition to these kinds of materials. So were there any surprises while you were putting this poster together, either the topic itself or just generally putting the poster together?
Not putting the poster together. I don't think so. I don't think I have anything to say about putting the poster together. It's a template that I've used before.
Okay. And
I try to lay out as many images as possible. I try to have them be as large as I can get them, because I think what I don't like are posters that really have these tiny thumbnail histology pictures. And I see these a lot at scientific meetings. And it does a disservice to all the hard work that goes into creating the section, photographing the section, or if you're using the slide scanner, which I think people are using a lot these days. I certainly have one in my lab that we use. So I wanted to give a sense of, like, here it is, look at it, get into the morphology, and actually be able to really see it well. So I guess the challenge is, technically, if it's a larger poster, how to construct this and keep everything aligned and make sure everything is color balanced properly. So those technical challenges of just constructing it, but actually doing the work to go into the poster is something a little different. So the technique of trying to work with these tissues are a little bit certainly more challenging than actually making up the poster.
Sure. And I have to say, I commend you for your image legend, because that is something always on every poster. It's the tiniest little thing to read. And what you really want is to know what all of those images are, and to have it, nicely standing out there to be able to read them and see them is a big deal. I think it's important, honestly. And I think you did a great job with that also.
Thank you.
It makes me want to be able to, look at every single photograph and see, and then be able to see everything. And it was very informative. As I said, a topic that I would never have thought I'd be reading. So it was very well done. And if someone is to walk away from your poster, what should they take away? Maybe in a few words, what do you think they should take away?
Well, again, many histologists that attend NSH are new to the profession. And even the ones that aren't new, that maybe mid-career or late career, that there are other options out there. There are other ways of doing things, other types of tissues that people look at and work with. And just have an appreciation for that and get excited about the potential of what histology can offer.
Mm-hmm.
Like I said, in the field of arachnology, morphology is only practiced by a few, and because again, depends on where you studied, who you studied under, what your mentor may be interested in. And it's hard to do, and I want people to take away that spiders are beautiful creatures. They have intricate minutiae of structures and things that you may not even considered. Who would have thought that a spider actually has teeth that are functional, that are used in a way that we use our own teeth?
Right.
No one would ever think about that. So I would like for people to come away with all of those points. Sounds
great. I think that, like you said, there's a lot of things out there that we do routinely every day, and it's
mm-hmm.
just is by rote. And when you find someone who presents a new topic or the ability to research a new technique, it just makes us much more energized when we're doing our work. And I think this is something that presents to techs that, hey, there's a lot out there that you can be involved with. And I think that was a really good thing that I took away from your poster. I thought that was just that kind of energy of, wow, look at that technique. That's just something I would have never thought. And that helps to inspire people, which I think this poster did. It inspired me, at least.
I'm very happy to hear that. Thank you so much.
You're welcome. If you were going to do something in a follow-up poster, what do you think you would do?
Well, I've certainly talked about fixatives before,
mm-hmm.
especially fixatives for a variety of arachnids. I'll probably continue on that line. I have a lot of things that I'd like to talk about, maybe obviously things that may not be applicable to a full workshop, but are more suited to a poster for technique. So I'd like to share things with people and have discussions. And maybe next year, if we can all meet in person, I'll have something else to show, and it'll be nice just to talk about it and engage.
No, I think that would be wonderful. And it's always nice to have that one-on-one conversation with people. So that's something to definitely look forward to for another time. It's a good venue for, like you said, something that may not be for a full workshop, but it does give some great information. We do love posters for that reason, and it gives people a chance to give some information that otherwise is not conducive to certain other presentations. So this works out very nicely. Well, anything else you'd like to tell us, Damien? Or I totally enjoyed speaking with you. If there's anything else you wanted to add.
Well, I'm more of a answer a question type of guy, so I don't want to get down the rambling road. But
no, that's quite all right.
I enjoyed putting it together. I certainly enjoyed talking about it. Yeah, I just appreciate the interest. So that's what I would have to ask. And if anyone is listening and wants to email me or has a question, they can certainly find my contact information through the member portal on
Great.
the website, and I would be more than happy to answer anyone's questions.
Fantastic. Well, Damien, it was a pleasure talking with you today, and I thank you very much.
Thank you very much, Mary. I appreciate it.
Thanks for listening to this episode of the 2020 Poster Podcast. Don't forget to check out NSH's other podcast series, including Histo Help and Women in Histology.